Cracking the Code of Alzheimer's. Revolutionary study unveils promising clue to predicting progression. Why is this study important, and what do you need to know in order to understand the implications? Alzheimer's disease is a condition without effective treatment options. The hallmark of Alzheimer's is cognitive impairment or decline. This is when a person has trouble remembering, learning new things, concentrating, or making decisions that affect their everyday life. It appears that people with such types of what we call mild cognitive impairment, or short MCI, are at higher risk of progressing to Alzheimer's disease than healthy elderly people. A biomarker is a substance present in blood or tissues, for example, that indicates the occurrence of a normal or abnormal process or the presence of a condition or disease. It can be used to predict how likely someone develops a condition such as Alzheimer's. Blood-based biomarkers have been identified as potential predictors of cognitive decline, which is instrumental for understanding what the research in the studies have tried to do. It all revolves around a brain structure called the hippocampus, which is known to be important for memory function. It turns out that it is highly sensitive to the circulatory environment, so the circulation provides blood supply to all the body tissue. Hippocampal neurogenesis, or in short HN, is the biological process underlying memory function. It happens in the brain of mammals. HN creates new brain cells, neurons, throughout our lives and is influenced by our activities and experiences. For example, interventions like exercise and diet that increase HN have been associated with decreased Alzheimer's disease risk. While some studies report a reduction in HN in Alzheimer's disease, there is a need for a better understanding of the changes in HN over time. Studying Alzheimer's disease has been challenging up to now because it affects the creation of new brain cells. Research has mainly relied on examining brain tissues after death. This limitation highlights the need for non-invasive methods that can allow research to study neurogenesis, so the formation of new brain cells, in living individuals with Alzheimer's disease. So imagine a world where we can anticipate the onslaught of cognitive decline before it takes hold. This study is our ticket to that world where we can hopefully one day pull back the curtain on Alzheimer's progression. What did the researchers look at, and what did they want to find out? The present study proposes an experiment in a lab, a so-called assay, that models the impact of the blood on HN using human cells that can transform into neurons, so-called hippocampal progenitor cells, and blood serum samples from people with MCI over time. They have conducted an experiment where they treated brain cells with blood taken from people who have MCI. The aim was to see how these brain cells changed and responded to the blood as Alzheimer's disease progressed, and determine whether the assay, so the lab experiment, could be used as a prognostic marker. What was done? This study was conducted by a team at the Institute of Psychiatry, Psychology and Neuroscience, IOPBN, at King's College London, and funded by the John and Lucille van Geest Foundation, the Medical Research Council UK, the Cohen Charitable Trust, and the Galen and Hilary Weston Foundation and the Rhodes Trust. Blood samples were collected from individuals who were initially diagnosed with MCI. Of the 56 individuals, 36 later developed dementia due to Alzheimer's disease, whilst 18 did not progress and remained cognitively stable. The researchers studied assay readouts and observed differences between blood serum samples from MCI converters, so those who had developed dementia due to Alzheimer's disease, and non-converters, so those who didn't actually develop Alzheimer's. Also, MCI converters completed significantly fewer years of education and scored significantly lower in the Mini Mental State Examination, the MMSE, a commonly used test for screening cognitive function, when they were compared to MCI non-converters. Different types of statistical regression analyses were used. A regression analysis is usually done for one of two purposes: to make predictions about the value of a certain thing we're interested in, based on some information we have about other things, or to figure out how one thing might affect another thing. But hold on, what did the researchers find? The baseline data from the assay, like a crystal clear crystal ball, predicted the trajectory for from mild cognitive impairment to Alzheimer's disease a whopping 3.5 years before official diagnosis. It's like having a time machine enabling us to glimpse into the future and potentially take proactive steps to protect our cognitive well-being. When they studied these samples, they found that the cells in the blood showed less growth and division, and more cells were dying through a process called apoptosis, which is programmed cell death. However, it was also noticed that certain substances in the blood caused more HN. So, is this the magical elixir that empowers the brain to fight back against the ravages of time? Well, we're not sure about this. More accurately, this finding suggests that the body may have a natural way of trying to compensate for the effects of aging or neurodegenerative conditions, which ultimately fails, so it isn't enough to halt cognitive decline altogether. The study suggests that blood serum from MCI converters may provide useful information for predicting the progression of Alzheimer's disease. Additionally, the study suggests that education attainment serves as a proxy of lifestyle that can affect the choice of occupation, socioeconomic status, and the degree of exposure to Alzheimer's risk factors throughout life. However, various lifestyle factors that may provide even better prediction of HN were not directly examined in this study. Are there any further studies needed, or is this ready to be used in real life? Well, the study suggests that we should dig deeper into how the substances in the blood, or more specifically certain proteins in the serum, affect brain cells in the hippocampus and the progression of Alzheimer's disease. This will help us understand the specific ways these proteins or substances work and their role in disease. But what does this all mean? The implications of the findings are that the method used in the study could predict progression from mild cognitive impairment to Alzheimer's disease up to 3.5 years before diagnosis. The study also suggests that increased neurogenesis, so development of brain cells, could be a compensatory mechanism in response to the aging or neurodegenerative processes happening in the illness. But it may not actually be a functioning restorative one that could halt cognitive decline altogether, as mentioned. To sum up, the researchers believe that these discoveries could mean that we can gain a better understanding of the changes that occur in the brain during the earliest stages of Alzheimer's disease. So now imagine a world where we can identify those at risk, intervene early, and rewrite the narrative of Alzheimer's. This study is essentially the roadmap guiding us towards a future where cognitive decline may one day become a battle we can win.